Hello everyone, I'm happy to present you today our work on unsupervised non-correspondence detection in medical images using an image registration CNN. My name is Julia Andresen. I work at the Institute of Medical Informatics in Lübeck, and this work is a cooperation with the colleagues in Kiel at the Department of Ophthalmology. I'm going to talk a lot about non-correspondences, so let me first clarify what I mean with non-correspondent image areas. For this, have a look at these two images from two patients where we can see several areas that I call non-correspondent. So on the one hand, we have inter-patient differences marked in red now. So here you can see a structure in the image of patient 2, but not for patient 1. And also we can see pathologies, a huge stroke lesion in the image of patient 1, but not visible for patient 2 here. So basically, these non-correspondent areas are image parts where the intensities between the images do not correspond, cannot be brought into accordance with each other. And if we now simply register these images, the results look kind of strange. So this would be an example of a CNN trained on these networks without taking on these images without taking the non-correspondences into account. Here you can see that we get implausible deformations where the background intensities are kind of pushed inside the brain simply to compensate the intensity differences that are due to the stroke lesion. Also, we can see that the structure that is visible for patient 2 gets kind of eliminated by our deformation. Again, an overcompensation of the intensity differences between the images. So this would be a behavior that we do not want to have, and this cannot only be observed for inter-patient registration, but also for intra-patient registration. So for example, if we look at changes over time, then again, we can see strange deformations here. So here you can see that the anatomy of the patient is kind of squeezed together, again, an overcompensation of the differences between the images. If we now use these deformation fields to estimate how much the patient progressed, how he's doing, then we would simply over or underestimate the progression, the changes. And this, of course, is pretty dangerous if we are looking at severe diseases. So what we want is a method that takes those non-correspondences into account explicitly. And this will now be the topic of my talk, how we achieve this. Taking a step back, the main goal of image registration always is to find a deformation that warps a moving image M such that it looks similar to the fixed image F. This deformation phi, for this example, might look like this here. So basically, we push the tissue upwards such that the upper border here gets aligned with the upper border in the fixed image. Such a deformation phi is typically found using this minimization problem here. So we are looking for a deformation phi that minimizes the image distance between the warped moving and the fixed image that makes these two images up here look similar, so to say. And the other component of this loss function is then the regularization of the deformation field phi. So basically we want that this deformation is smooth. Now you can see that here again we have non-correspondent image areas highlighted in blue. And if we now perform a subtraction, if we calculate the difference image between the two images, we can see that the result looks like this. And exactly in these non-correspondent areas, we can see large differences between the two images. And this could now be used by maybe simple thresholding to generate a segmentation of these non-correspondent areas. But as we have seen in the examples earlier, this registration process without having such a segmentation can lead to large errors, to really implausible deformations. So basically, we do not want to do this in the order I just described, but we want to find the segmentation during the registration process. So we changed our minimization problem as follows here. We do not only search for a deformation phi now, but also for the segmentation S. And we mask out our image distance measure exactly in those regions that are segmented. So basically, we want to find the image areas where the image distance is especially large, so where we have those huge differences we saw earlier. If we now optimize this problem as it stands here, we would get a very trivial solution we do not want. So basically, our network would segment the entire images simply to mask out the image distance at everywhere. So we add these two loss terms here, the volume and the perimeter of our segmentation, that favor in the end that the segmentation we get is small only and that the borders of the segmentation are smooth. This is not a new idea. It was presented by Kang-Ling Chen and his colleagues a few years ago, but we've now been the first ones to take this approach and to transfer it with slight modifications to the field of deep learning. So we are not using an iterative optimization approach here, but we use this training objective to train our CNN. And our CNN is called the NCRNet, the Joint Non-Correspondence Segmentation and Image Registration Network. And the basic structure can now be seen here. So input are the moving and the fixed images. They're passed through one encoder, and we then have two separate decoders that output the diffeomorphic deformation field phi and the segmentation of non-correspondence as S out of the second decoder. One important thing to note here that is described further in our paper that we published in the International Journal of Computer-Assisted Radiology and Surgery in this year is that the outputs are not only produced here at the end, but also at some intermediate steps of the network to produce outputs for 3 levels of resolution. And for all 3 levels, we then optimize the training objective simultaneously. So basically, we are doing the optimization for all 3 resolution levels, which in iterative approaches earlier would have been done in successive order. This network is trained in a 2-step training procedure. So first we perform a pre-training using the kind of classical image registration objectives, that is the image distance, the regularization of the deformation field, and here at the end we add some weak supervision to the registration task. So we are calculating the overlap of the segmentations of the moving and fixed images. Here we use not any fine-grained segmentations, but rather segmentations of the huge anatomy that can be seen in the images. So for the OCT examples here, we would use the segmentations of the entire retina, marked here in orange. The second training step then updates the entire network using the loss function I presented earlier with this masked image distance, regularization of the deformation and of the segmentation. And again, we add this weak supervision here. This gives us pretty good results. So for comparison, I again repeated the image we saw earlier with the strange deformations given by the network that does not take the non-correspondences into account. And down here, you can see the results of NCRNet. And you will see that the deformation we get is much more plausible, and also the structures that are only visible for patient 2 are retained much better here. So in the end, we get a registration that is much more robust to even large lesions, large pathologies. We performed several experiments with this network, and the first thing we did is some ablation studies where we compared a fully unsupervised version of our network to the weakly supervised version, where we used the dice score and the loss function. So we trained it once with and once without dice loss in the training objective. The second ablation is comparing the 2-step training procedure to training the network with the full loss function during the entire training process. And we compared results then based on the OCT images and the 3 anatomical labels that are marked here with different colors. And we calculated the average symmetric surface distance for these markers here. So lots of numbers. I want to highlight 2 main results here. The first thing is a comparison between the supervised and the unsupervised network. And here you can see that the results are almost comparable. So the cool result we got about from this ablation study is that you can use this network on any data set, even on data sets where you do not have any manual labels, and you will still get very good registration results, which is pretty good. The next thing I want to highlight is the comparison between the 2-step training and the full loss function during the whole training process. And here you can see that the 2-step training really significantly improves the results. So here, if you would like to use this method, I really recommend to perform this pre-training of the deformation decoder. A second experiment we performed is to use the segmentation output of the non-correspondences given by our network to directly segment lesions, pathologies in our images. So here's an example of a patient with age-related macular degeneration, where he has, in the one time point, he has a huge lesion visible, and in the other time point not. And if we now use our network to segment And to register these images and to segment the non-correspondences, you will see that the non-correspondence segmentation directly aligns with this lesion that is only visible on one of the two images. So in such a case, the network can directly be used to segment pathologies in a fully unsupervised manner. And for interpatient registration, you will see that here we get a segmentation of the lesion, but also the interpatient differences are highlighted by our network. So here we cannot use the segmentations directly, but with some weak supervision using region growing inside our lesion, we can still get very good segmentation results in the end that we also compare to some other state-of-the-art methods to do this. And we could show that we outperform this method. To summarize now, we presented a joint non-correspondence detection and image registration framework that achieves state-of-the-art registration results. So that is another experiment that you can read in our paper. And the cool thing about this registration is that it's really robust to large lesions and that you can use the resulting segmentations also to perform weakly supervised or fully unsupervised pathology segmentation. One last thing in this talk here is a small outlook on our current research. So we extended this framework to not only segment the new lesions, but also to model the intensity differences between the non-correspondence areas. So basically, we do not only segment them, but we model also what is different between the time points or between the patients. And this is an application to multiple sclerosis where we segment new lesions. And here we can now analyze the morphology and the intensity profile of these new lesions, which gives us interesting medical implications. I end this talk with a small outlook, and I hope you enjoyed it, and I'm looking forward to your questions. And yeah. Feel free to contact me via email. Thank you for listening.